So KRAS mutation is the most commonly mutated oncogene in cancer. It affects roughly 30% of non-small cell lung cancer in particular. And with this work that we've seen developing, it's a G12C mutation that's common to lung cancer where we've seen inroads. Before we think about this progress, I think the context of it needs to be recognised in terms of the difficulty that we've had in the past. The mutation was discovered in 1983 and has been considered for over 30 years to be undruggable. And if you compare with BRAF mutation, for instance, it was recognised in roughly, I think, 2002 and had a tractable treatment available in 2009. So a development that only took 7 to 8 years at the time. This progress has really been a momentous shift into terming KRAS mutant cancer as something that is druggable rather than undruggable. The clinical trial disappointments with KRAS mutant targeting in the past can probably be looked at in particular in 3 senses. First of all, there was the introduction of farnesyltransferase inhibitors with trials around 10 to 15 years ago, which were disappointments. The biological basis for this was that KRAS was thought to be dependent on farnesyltransferase for translocation from the cancer cell cytoplasm into the plasma membrane in order to become active. Turned out this wasn't the case, and that's some of the reason why these clinical trials were a disappointment. And these drugs have actually made a revival recently in HRAS mutant cancer, which does happen to be dependent on farnesylation. The second disappointment occurred with MEK inhibition. And MEK's a protein immediately downstream to KRAS mutation. So again, a sound biological basis for trials such as the SELECT-1 phase 3 trial. But again, this trial was, I think, a big disappointment at the time. Part of the reason for this was there were very promising randomized phase 2 results suggesting that this would work, emphasizing the importance of, at least in this setting, performing large-scale randomized phase 3 studies. Some of the other reason why this failed was really just down to the biological rewiring of KRAS mutant cancer in the setting of MEK inhibition. And the last emerging step, which I think at least has been a disappointment so far, is this concept of synthetic lethality, with the main synthetic lethality target with KRAS mutation postulated as CDK4. We've had trials now looking at inhibition of this protein in KRAS mutant cancers, such as a phase 3 KRAS-selected Juniper study. And the results haven't been promising so far, suggesting that these drugs would at least need to be combined with something else. And that immediately brings into play the possibility of toxicity concerns in frail patients.